He's studying the diversity and biology of helium-4, which is what he's going to talk to us about tonight. He's a systematic biologist who incorporates data from multiple disciplines, such as morphology, chemistry, and genetics, to better understand his study species. He will present on his recently published study of the leaf types, flowering, and soil science of the almost extinct Sarracenia jonesii, his push to encourage a better understanding of the soils carnivorous plants inhabit, and his current doctoral dissertation on helium-4. After his presentation, I do have a slide with his Instagram and TikTok account, so you can tag along with his academic journey. He's also done some talks with the International Carnivorous Plant Society, which are on YouTube, so you guys can follow up with that if you'd like. All right, Joseph, I'm going to hand it over to you and scooch it over. We do have speakers throughout the room, so theoretically, you may be able to hear questions as you go, or if you want us to hold those to the end, just let us know. And welcome.
Awesome. Thank you. Yeah, we can do questions at the end if that works. And let's see, can you see my screen now? Hopefully, is it full screen? Yes. Okay. I'm going to take that as a yes, hopefully.
Yes, yes.
Okay, great. Thank you. Okay. Well, yes, thank you for the introduction. I was at your meeting, I think was it May? And so I'm excited to finally present for you. And so, as Jess said, I'm here to talk about morphology, taxonomy, and soil of Sarraceniaceae. Um, and so I wish I could be there in person, but actually Madison and St. Louis don't have a direct flight, and that's really annoying because like when I went there, I had to do like a 4 hour layover in Dallas, and that was frustrating. Anyway, so a more introduction about me. I started out my plant journey growing plants just for fun. And I particularly enjoy growing Echeveria, which is a genus of succulents, as well as carnivorous plants, of course, and some really frustrating high humidity plants that like to die when you just look away from them. This is an orchid I helped describe, like a new species, and some microscopy I did as evidence that I like plants that aren't just carnivorous. But my other plant identity is as a plant scientist. I got my bachelor's in environmental studies, my master's in botany, and I'm currently getting my botany PhD. And my PhD is on Helianthera specifically, their evolution and morphology. And also starting next month, I'll be in New York City as a research assistant at the New York Botanical Garden, working on completely different plant science stuff. And so I like to be specific about the kind of botany I do. Specifically, I do systematics, which is the scientific study of the kinds and diversity of organisms and of any and all relationships among them. So specifically, just how diverse these group of pitcher plants are is kind of a broad way to describe what I do. And taxonomy, which is a little piece of systematics, here's a nice quote I like that it's a synthetic science drawing upon data from such diverse fields it has no data of its own, meaning that it uses and incorporates lots of different tools and techniques. Like here, I'm showing microscopy and genetic DNA work. And so when I was there in May, you guys had a great talk about Sarracenia taxonomy, basically. So I was really happy to hear that like you have a precursor already. And so this is kind of building on that as well. And so I do a lot of different kinds of work. These days, it's pretty much entirely lab and collections based, but I have done and do some field work, and I'll show you that as well. But my interests definitely lie in collections-based research, which we'll talk about later on. And so, if you aren't familiar with the term, Sarraceniaceae is the family of the American pitcher plants. We have Sarracenia and Helianthera, which are sisters. They're more closely related to each other than they are to Darlingtonia. So technically, I won't be talking about Darlingtonia today because I don't study it, but as you know, it's a wonderful species. And so Sarraceniaceae is really just the umbrella term for all of these three genera. And so the first part of the presentation, I'll be talking about evaluating endangered pitcher plant leaf morphology and flowering as related to soil nutrient status, specifically for Sarracenia jonesii. So Sarracenia jonesii, which some consider to be a rubra subspecies, is a wonderful clumping pitcher plant in the southern Appalachia. It's specifically native to North and South Carolina and only grows in these specific habitats called cataract bogs and fens. It's a rare and federally endangered species. It's almost extinct. And because of that, it's also very poorly studied. And so there's been an overall population decline observed since humans first started recording this species. And it's been extirpated from 43% of its known populations. And extirpated, it just means extinct in those areas. And so in 43% of its known range, it no longer lives there. So that's actually a really, really big deal. That's a huge, it's like half of its range gone. So it only exists now in 12 known populations. And so, of course, there's a lot of conservation work that is done in those 12 populations. But if you're familiar, Sarracenia hybridize really well, and that causes a lot of problems with their conservation. So I studied one of these 12 populations at Roberts Nature Preserve. And so the preserve is the meta population overall. And then within that meta population, there were two subpopulations and then one lone individual. And so I studied all three of those. And historical reports shared that there was a fourth subpopulation, but myself and other botanists looked, and we just could not find it. And I do believe that it was extirpated, even though this is a greatly preserved area. And so I know I haven't really talked about the work I've done yet, but let me just give you a few definitions before we start. For this work, defined a clump as a group of individuals distinctly grouped together, likely sharing rhizomes. If you've ever repotted a Sarracenia pitcher plant, you're very familiar that, within a big pot, you could really kind of take apart multiple individuals and put them into multiple pots. But for my purposes, a clump like this served as one clump, one individual, essentially. And so, just a little precursor into the morphology. Of course, the famous pitcher is the carnivorous leaf that eats their prey. And then there's also Phyllodia, Phyllotium in the singular, and these are the less well-known non-carnivorous leaves in Sarracenia. And they are actually better at photosynthesizing because they don't have all of this carnivorous chemistry in the way. But they're more expensive for the plant to produce. It takes more resources and energy. And then, of course, the flower. And for my purposes for this study, it didn't matter if the flower was fresh or dried. And so I really won't get into my lab or statistical methods, but for my fieldwork methods, I randomly selected clumps, and at each clump, I counted the number of pitchers, which I will never ever do that again. That was horrible. That was counting thousands and thousands of leaves in the direct sun. I hated that so much. But it was a learning experience. And then I counted the number of Phyllodia, the non-carnivorous leaves, the number of flowers. I took a clump circumference, and then I also took these soil cores so that I could analyze the soil of the plants that the soil the plants grow in. And so I had four main hypotheses, and I'm going to go one by one. My first one was that more Phyllodia, more of these non-carnivorous leaves, will be found in the clumps with the most plant available nutrients in their soil. I'm going to talk more about the term plant available nutrients in a few minutes, but bear with me for now. And so I had hypothesized this because we see in experimental, like kind of lab-based studies, that when you fertilize Sarracenia, they grow more of these non-carnivorous leaves because the thinking is that if the plant is getting more nutrients from their roots, they don't really need to eat as many bugs, so don't need to grow as many carnivorous leaves. But ultimately, I did not see that relationship. And that's likely because in a healthy fen system, like a natural healthy habitat, you're not going to see as many nutrients in the soil as you would in these experimental soils where you're fertilizing them. And so I promise there won't be too many graphs because who wants to look at graphs at 7 p.m. In the evening? But I will walk you through them so it won't be too bad. But this one's just showing there's not really a relationship between plant available nutrients and these non-carnivorous leaves. And I also didn't find that relationship with pitchers. And so this was a dud. And then the next hypothesis, this is where we'll talk about plant available. So I hypothesize that soil in which Sarracenia jonesii grows in is not nutrient poor, but rather nutrient rich, meaning a high total nitrogen. But these nutrients are not in plant available forms. So I'm sure you've heard and read and probably used the term nutrient poor to describe the soils and the habitats that carnivorous plants grow in because we're always explaining the story that that's why they need to eat bugs, and that's true. But I am hypothesizing that the soils are not actually nutrient poor in the sense that they're lacking them and that they're actually high in nutrients, but those nutrients are chemically in a form that the plant can't take up. And so nutrients can just be chemically in a form that the plant can take up. They're not dissolved in water, and they're sitting there and just kind of not being used by the plant. Think of your compost, where if you put a banana in, eventually it'll become a healthy soil, part of soil, right? But there's a process where that has to happen. And that process where something turns from organic matter, like a banana peel, all the way to healthy soil is decomposition. Because decomposition will break down that organic matter into its basic building blocks, the elemental components that another organism then can use. But in order for decomposition, this important process to happen, there are multiple things that have to be in the right order for it to happen properly. For example, if the environment is very acidic, there's not going to be as much decomposition happening. And if the environment is very wet, very waterlogged, there's also not going to be as much decomposition happening. So if you're familiar with carnivorous plant soils and habitats, even when you grow them, in your pots, they're very wet and acidic soils. So I'm hypothesizing that there's actually going to be a lot of undecomposed organic matter. And that's actually because the little microorganisms that do the decomposition, most of them want environments high in oxygen. But when the soils are filled with water, there's not a lot of oxygen. So there's much fewer little microorganisms that can do the decomposing. And so ultimately, that is what I found, that the soil in all the subpopulations had a relatively high total percentage of total nitrogen. That means a lot of organic matter, but low amounts of nitrate and ammonium. Here, nitrate and ammonium being the plant available nutrients. And so actually, we're going to ignore that graph. I thought I deleted that. So here we're looking at this table. These are levels of what I would call a normal soil. So a soil that, doesn't have carnivorous plants versus what I found with the soil that has the carnivorous plant. So percent N being the total nitrogen, the like organic matter, a normal soil would have 0.1 to 0.15% total N, but I found that this soil had zero point 0.2. That's a lot higher on this scale. And then these two are the plant available. And so I thought that the carnivorous plant soil would have less, and sure enough, these have quite a bit less. For example, 2 to 10 parts per million of ammonium, and the mean of the carnivorous plant soils was 0.5. So this is showing that there was a lot, in fact, more nutrients than a normal soil, but they're just not in a plant available form. And so I go back to the idea that a lot of us call these nutrient poor soils. And that is true in the sense that it's functionally nutrient poor, right? It's as if the soils did not have nutrients. But in fact, it's just that those nutrients are plant unavailable. And so this might seem like semantics, and it is to a bit, but also at the same time, it's biologically and chemically very different because these soils actually do have a lot of nutrients. And if something about the habitat changed, like maybe the soils became less acidic or less waterlogged, maybe those nutrients would later become plant available. And so if more people do more studies of these different carnivorous plant habitats and see the same thing, maybe we can start to use a term more like plant available or unavailable rather than nutrient poor. Because I hypothesize this would be true for pretty much every carnivorous plant habitat. And then the third hypothesis was that the more plant available nutrients that nitrate and ammonium were in the soil, the fewer flowers the clumps would produce. And I base this hypothesis based off of experimental studies showing that when you fertilize using the soil, Sarracenia, they're going to flower less, probably because the plant is not used to that amount of nutrients in the soil and they're not that happy. But I did not find that as a relationship, but more data might have shown it. And so, what I did find that was interesting was that there was a statistically significant relationship that the more pitchers one of these clumps grew, the more flowers it grew. And so, this makes sense if you think about the more pitchers a plant has, potentially the more bugs it can eat. The more bugs it can eat, the more energy it has. So, maybe it can grow more flowers. So, that was an interesting kind of find on the side. And then my final hypothesis for this project was that as one of the few extant sites, which means not extinct, as one of the few not extinct sites, I hypothesized that the Sarracenia jonesii metapopulation, the overall population at the preserve, would show declining trends in population growth since 1979. And that might seem like a random year, but that's just because that's the first year that we had historical data for the site. And so, sure enough, as we see across the whole species overall that I mentioned at the beginning of the presentation, the metapopulation is slightly declining in numbers, or in number of clumps. But interestingly, the clumps are growing more leaves and flowers than in the past. And I'm guessing that's because of these increased conservation efforts, but then also increased maturity, likely an older individual, because these plants can live decades and decades. Probably an older individual is going to grow more leaves and more flowers. And so, how is this going to be applied? I hope that conservation work can use this information on what the soil of the native habitat of this very rare species is like so that they can kind of use the ideal soil conditions. And the soils of this very specific type of habitat, Southern Appalachian cataract fens and bogs, is very understudied. We don't really understand these habitats as well as others. So learning anything like this can be valuable. And that, I was able to show the nature preserve owners that they need to continue to invest in conservation strategies and monitor the populations. Because although the plants may be growing more leaves and flowers, the actual number of clumps is slightly declining. And so, again, going back to the whole soil thing, which I know I talked a lot about that, but I think that's like the most fun part, is that maybe with more similar studies on other carnivorous plants, we can move away from the nutrient poor label because I think it's a misnomer when really there's a lot of nutrients, they're just not plant available. And if you're interested in reading more about this and actually how I did the chemistry work and the statistics and whatnot, I have a paper out about it. And it even made the journal, the cover of the journal, where you can see this is a picture I took of a praying mantis stealing food out of the pitcher, which is something they do, and it's so rude to the plant. But I guess the praying mantis needs to eat too. And so now we're going to move to the other part of the presentation. Going back to the umbrella of Sarraceniaceae, we're going to move away from Sarracenia and now talk about Helianthera, which is the work I do day to day now. So this part of the presentation is called When Damage Becomes Diagnostic, Cracked herbarium specimens reveal a new taxonomic character in Helianthera. And it's okay if some of those words are unfamiliar. I'm going to walk through it. And you'll notice it's like a completely different kind of techniques and tools for this work. And so Helianthera, as I mentioned at the beginning, is the South American group of pitcher plants, one of the three genera in Sarraceniaceae, and it grows in Venezuela, Brazil, and Guyana on the top and on the sides of these very unique habitats called the tepuis. These are sandstone plateaus, and remain one of the least explored and least understood habitats on the entire planet Earth. So this is a very unique environment. And they have these upright, erect tubular leaves that act as a pitfall trap. And what's really noticeable about them are these small appendages at the top of the leaves that have a high concentration of extrafloral nectaries that we commonly refer to as a nectar spoon. And it's there for prey attraction. And so, if you step back and look at the genus as a whole, there are 24 species and one subspecies. And personally, I think they all kind of look alike. There are certainly differences between all of them, but it took me a while to start to really instinctually be able to tell them apart. And so part of my PhD work, I wanted to figure out, can I find ways to differentiate these species even more? Because if you look too quickly, they're all just kind of red and green tubes, basically. So to do that, I went to trichomes in carnivorous plants. Oh my gosh, has this been in the way the whole time? Hopefully not. Anyway, and so trichomes are these epidermal outgrowths that are either uni or multicellular, and they're made of the epidermis of the skin of plants, and they have multiple different uses across carnivorous plants. Most famously, they are the triggers in the Venus's flytrap. But as you can see in these examples, they have lots of different functions and are found in so many carnivorous plants. And so I set out to do a micromorphological survey of the whole genus, which meant that I wanted to look at every part of the leaf of every species in the genus so that I could kind of look at them as a whole and see, are there any differences? And if so, what are they? So I used living samples from private horticultural collections. And I had all species represented and multiple potential new species as well. And so, in order to realistically do this project, I had to separate the leaf into four zones. This is zone one, two, three, and four. And then from each zone, I would cut out a little piece of tissue, and that is actually what I would image. And I would image both sides of it with an electron microscope. And so this part of the presentation is actually not about that work at all. It's talking about what I did with it. But so here are some results from it. This is the top inside of a pitcher of three different species. You can see this one doesn't have any trichomes, which is what we would call glabrous. I don't like to say hairless because this isn't hair. Only mammals really have hair. And then other species do have these trichomes. Some might be a little bit more sparse, some may be more dense. This one looks like it's a little bit more wide. And then in the bottom outside, so the bottom of the picture on the outside, these are bifurcated trichomes, meaning two-sided. Some can be quite wavy, some can be straight, and some can be non-existent. And then here we are on the bottom inside of the pitcher, and some can be somewhat long, some can be extra long, some can be shorter and squatter. And so when I looked at all of these hundreds of photos across the whole genus, it was here in the bottom inside of the pitcher that I noticed the most dramatic differences in the trichome form. So it was here that I thought I could kind of tell a story, but I needed to investigate it more. And I was really curious, why did the trichomes differ so much? But if you recall, I was using one leaf or a handful of leaves really from each species from a living collection, but I needed a bigger data set. So then I headed to herbaria. And so what is an herbarium or herbaria in the plural? It is a museum collection of preserved pressed plants that are used for scientific research. And the specimens act as a biodiversity library representing the natural history of the earth. And so every single specimen acts as a little point in time and space saying, hey, this organism was here right now in this point in history. And there's other information that you add along with the specimen, and they're extremely valuable. And most importantly, it allows me to study Helianthera outside of their extremely inaccessible habitat. And of course, herbarium specimens are essential for, all plants, not just this one cryptic group that I study. And so here are some images of what herbarium collections look like. Someone told me today it looks like the movie Backrooms. I haven't seen it, but it definitely can be kind of creepy when the lights are off. They'll often have the lights off to preserve energy, and the plants are dead. They don't they don't need the lights on. But these are multiple different institutions, so you can see kind of what they look like. Herbaria are typically not open to the public. They are safeguarded, protected, natural history collections that require credentials to go to. But this is why I was in St. Louis 2 weeks ago, was to go to the Missouri Botanical Garden Herbarium, and it perfectly lined up with your meeting. So hopefully some of you in the audience today I got to meet. And this is what it looks like inside one of the cabinets, for example. It's folders of dried pressed specimens. And so I was really curious about the bottom inside of the picture, so I planned a bunch of travel. I first started out at my home herbarium, the Wisconsin State Herbarium. I looked at the Helianthera specimens there, and then I went to the North Carolina Botanical Garden Herbarium, looked at their specimens. Then I went to New York City to the New York Botanical Garden. They have what I am hypothesizing to be the largest collection of herbarium specimens of Helianthera in the world. And then I went to, so, and NYBG is one of the largest herbaria in the world. And then I went to the Field Museum of Natural History in Chicago, which also has one of the largest herbaria in the world. And then I went to St. Louis, and Missouri Botanical Garden also has one of the largest herbaria in the world. And in fact, it takes up two huge buildings on their campus. And then just yesterday, I got back, ironically, from the United States National Herbarium from DC in their National Museum of Natural History. And so this travel allowed me to go to some really gorgeous buildings. And so it's sometimes I like to expose people to the idea of these natural history collections, because you might think, as a scientist, a biologist, as someone who's digging in the ground all day. And while that digging and trekking through the forest is what created these specimens, this also allows us to now look at these specimens even decades, hundreds of years later, in these really pretty buildings. And so many herbaria across the US had maybe just a handful of Helianthera specimens, like maybe 1 to 5 specimens. So it didn't justify a visit, but they were able to loan me the specimens and send them in the mail. So I made a map of like where the various Helianthera specimens came from. And so to be clear, though, this is not a map of where those plants grew, right? They all came from Northern South America. This is just a map to show you where the collections were housed, because I thought that was kind of cool. And so once I sit with the specimens, the first complication I had was that I could not alter or damage the specimens, but I needed to see the interior of the picture. Technically, you can request to damage specimens. It's called destructive sampling, but it's a long process to request that. A lot of the times they don't get accepted, or you have to have a really good justification, right? And sometimes you're lucky enough that a Helianthera specimen is kind of pressed open so that the interior can be viewed, but those are not that common. This is an example of one. Whereas I find that a lot of them look more like this, where they're kind of pressed, normal or closed. And then I realized that I can view the actual interior of the picture through cracks in the epidermis of the picture. And this is the really exciting part, where I figure out I can see the interior of the pictures without damaging the specimens. And so if you've ever felt or held or maybe broken a Helianthera leaf, they're very rigid and very brittle. I like to say that Sarracenia bends, but Helianthera breaks. And so when they're pressed, they usually have these dramatic cracks that go across the length of their body. And I realized I can use these to my benefit to view the interior. And it's ironic because when pressing herbarium specimens, you typically don't want cracks, you don't want damage. You want to represent the plant as best you can as it looked alive. But with most plants, they're not these 3D tubular weird shapes, right? There's like a front and a back of the leaf. And in those cases, then they press, a leaf that's showing the front of the leaf and a leaf that's showing the back of the leaf. But again, with pictures, it's this kind of weird situation. And so to show you or to represent the prevalence of these specimens with cracks, out of all the specimens I looked at in New York, 92% of them had cracks, showing that I could view the interior of almost every specimen without damaging it, which was great. And so, if you recall, I'm really interested in the bottom inside. So I would pick up a specimen, I'd look for a crack, and then I'd zoom in even more under a microscope, and boom, I can see interior trichomes. And so, if I were to put this specimen under the electron microscope, this is what those trichomes would have looked like. And so I did this, but then hundreds of times across all the different various species that were represented in these herbarium specimens. And so this is a callback to the micromorphological survey I did with living specimens, where I saw these different types of trichomes. Sure enough, I saw these in the herbarium specimens, and I was able to categorize all species into two types of bottom inside trichomes. Some species have trichomes that are short and compact, and they're kind of squat and have these big round bases. So this is a photo from an herbarium specimen on the top, and this is an image from a scanning electron microscope on the bottom. And then the other species have long and thin trichomes. Here's one that is successfully holding onto an ant. And again, here's an image from an electron microscope. So you can tell that these trichomes, they're in the same area of the leaf, but on different species, they're dramatically different, what we call morphotypes, meaning they're different types of this structure morphologically. And so I dubbed the short and compact ones as C trichomes and the long and thin ones as LT trichomes, and introduced this as a new taxonomic character. And so that means it's a characteristic that it can be used to distinguish and classify organisms from each other. And so this is a list of Helianthera specimens that are species that have SC trichomes and ones with LT trichomes. And once you put these onto the tree of life to see how these species have evolved, there is a story that the ones that have SC trichomes evolved together and the ones that have LT trichomes evolved together. And so this opens up a can of worms to ask questions like why do they have these different types of trichomes? Why are they arguably in the least accessible part of the leaf, all the way at the bottom, under the water level? There's so many questions to ask, and now that we know this. And then you might have also been asking yourself, we already can identify Helianthera, most notably with this taxonomic character, the nectar spoon. Why did you do all this work to find another? And that's because species names, what we consider a species for all organisms, not just pitcher plants, they're hypotheses because we're trying to categorize something that's on an evolutionary spectrum. So we're always trying to collect more data to see how do these organisms differ if they do at all. And the better that we can differentiate, classify, and understand these organisms, the better we can hypothesize how they evolved and their place on the tree of life. And the more we know about them, the better we can protect them. Because taxonomy and classifying organisms is essential to protecting organisms and their legal protections. And interestingly, Helianthera might be one of the most protected groups of carnivorous plants because they grow somewhere that's so incredibly inaccessible. But as we've seen time and time again, humans will always find a way to reach something, and so that may not last forever. So we need to learn about these organisms. And all species of Helianthera were described using a morphological species concept, meaning that when they figured out which species were which, they only did it with morphology, not anything like genetics. So that means morphology is really important to the identification and identity of Helianthera. And so this is an illustration from when George Bentham described the first species of Helianthera in 1840, which was Helianthera nutans. And I like to point out that they drew a short and compact trichome. So it's the oldest known illustration of a Helianthera trichome. And there they are, these little short and compact ones at the bottom of the picture. And so if you're interested to learn more about this taxonomic character and which species have which and how I figured it out, I have an open access paper, which means it's free to read and download. And it was written with a broad audience in mind. It's not super technical. It's not supposed to be confusing. And it's short. It's like 4 pages, and it's got a couple more figures that I didn't show here. And so even if you just Google when damage becomes diagnostic, it should pop up. But there's also a QR code here in case you want to access it, or you can always reach out to me. But let's see how I'm doing on time. I'm doing perfect on time. So there's a lot of folks I want to thank. So a lot the work I presented today was from both before my PhD and during it. And so a lot of funding went into funding this work. And then part of the reason I'm here is also because you guys were so gracious and kind to donate to my work as well. And so the work that you donated to is on my molecular work, rebuilding the tree of life for this group. And I'm currently writing it up. I'm currently working on it. And so that's not what I presented on today because it's not done. But know that it went and to something you good and it was used. And I'm excited to get that one kind of wrapped up and out the door. And I also absolutely love your logo because Helianthera is just right there, right in the middle. That's how it should always be, right? And I saw that I think you guys got a new logo, but Jess, if you still have shirts with that logo, please do let me know because I would definitely buy one. And then I didn't do the science alone. I've had my advisor, and then I also have a fantastic group of undergraduates I call the Pitcher Pals, who help me with specimen preparation and all sorts of great stuff. And then my kind of final remarks is that science is struggling across the country, but botany is also suffering from this. This is an interesting article called "Are Botanists Endangered?" where she describes that the number of botanists is declining rapidly, and the people filling those spaces are not botanists, and the drop in research funding for pure botany is has tanked in programs across the country, both at the federal level and not. And it's actually quite cool that you guys are so close to one of the botanical powerhouses in the entire world. The Missouri Botanic Garden is really a world leader in botany, and so it's quite cool that you live right there. But make sure to stay curious and engaged because scientific support is so essential for society and mitigating climate change, and everything goes back to plants, even our medicine. So it's really important that we continue to protect science. And apparently, Jess, I think also has a slide, but I have an Instagram here that you can follow. That's where I mainly post. I have a TikTok as well, and I'm pretty good with DMs if you ever want to DM me, but you can also reach out to me with email. And yeah, that's it. So I can take questions.
There are speakers around the room, so if you're loud and clear, he should be able to hear your question. We can talk into the microphone if you have a
Perfect.
hard to hear.
But if no one has a question, that's also okay. Gorgeous. What's the other plant growing with it? A begonia? Okay. Which species is it?
It's actually, I almost wanted to ask you to guess, but the lighting's terrible. You can
take a photo and send it afterwards.
It's a hybrid.
It's a hybrid. Okay, that's what I thought. Is it with Pulcella or something?
Yes, Pulcella by Meyer.
Okay, cool. That's
Like
awesome.
that crusty, tiny, dingly video.
Okay, nice.
Tough.
Are you the one that did the Sarracenia presentation?
Yeah, the one in the perfect.
Oh, that's awesome. I'm glad I was there for it.
Thank you so much, Joseph. That was
Thank
really
you.
enjoyable.
I guess
Wonderful.
One question. So I know it's ahead of publication, but is there any helium flora genetic data that's going to rock my world? I can feel it now.
I always think of course it will rock your world. Yeah. There uh yeah, it's pretty impressive what we've been able to do with just morphology. And so I'm both grateful to be able to provide insight into new relationships and confirm relationships with the genetic data now. And as you may know, it's extremely expensive. So that's why your donation was very helpful. Yay.
For taxonomy, at least from the experience I've had, it's fairly recent. And usually it's been involved in plants switching genuses or subspecies or whatever. So in the Helioflora that you looked at, did anything change from one genus to another?
That's a good question. No, Sarraceniaceae has been pretty constant in which plants are in which genus. And that's largely driven by geography because they're all so disjunct from each other. And so, meaning that we know there's no Sarracenia in South America and vice versa. But it is exciting that we have molecular data to show that Sarracenia and Helianthera are sisters, so they're more closely related than Darlingtonia is to either of them. And that's a really weird story because you would think that Sarracenia and Darlingtonia being on the same continent would be more closely related, but that's still a mystery to find out. Good question.
Question you brought up earlier with your nutrient talk in the soil biomes. Do you think that the changes in temperature and these kind of effects of climate change are going to have a combined effect with that to change these populations drastically? Is there a further inference of that you can make?
Yeah, that is definitely one of the dangers because climate change has been and will continue to change soil chemistry. And what I think will be even more pressing is that with more climate change, we're going to have more invasive species. And carnivorous plants are famously poor competitors. So the more invasive species you have, the more competition there'll be, and they'll kill out the carnivorous plants. And so, yeah, there's multiple pressures on carnivorous plants from climate change from multiple different angles. But I do anticipate the soil chemistry to change. And then also, if you think about, the more we fertilize, the more kind of chemicals we use in the field that can cause runoff, that can change the soil chemistry around these plants, independent of climate change as well. Awesome. Well, thank you all.
Thank you very much.
Thank you.
Yay. Okay. Goodbye, everyone.
That was really cool. All right.